Good afternoon, everyone. Thank you for joining today's webinar, Community Information on the Hepatitis B Cure. My name is Catherine Freeland, and I'm the Public Health Program Director at the Hepatitis B Foundation. I'm pleased to present with my colleagues at the Hepatitis B Foundation and ICE-HBV today's webinar. Today, we will learn about progress towards a hepatitis B cure, and we want to welcome you to today's session. Before we get started, I would like to go over a few information on how to participate in today's webinar. Please note that all attendees are in listen-only mode and will be muted during the presentation. There is a phone-in option for the United States that's listed here. If you need alternative country phone lines, you can go to the GoToWebinar panel. You may submit questions in writing during the presentation by typing them in the box on your GoToWebinar control panel. We will be reviewing questions as they come in and hold a Q&A period after all of the presentations for today. And finally, today's webinar is being recorded. You will receive an email with a link to view a recording of today's presentation after the session. We would like to start out today's session by sharing with you a short video. And it looks like that technology is not going to work for us very well today, so we'll be sure to send that out in the recording. I apologize for that inconvenience. So we'll proceed. I'd like to start out by introducing you to our esteemed panel of speakers for today. Our first speaker today is Dr. John Tavis. Dr. Tavis received his PhD in molecular biology from Pennsylvania State in 1990 and postdoctoral studies with Dr. Don Gam at the University of California, San Francisco. Since 1994, Dr. Tavis has been an investigator at St. Louis University School of Medicine. Currently, as a professor of biology, his lab focuses on HBV and has since inception, and is currently pursuing basic biochemistry and antiviral drug discovery targeting the viral ribonuclease H. Additional interests include HCV genetics and anti-herpes virus drug discovery. Dr. Tavis has served on the American Cancer Society peer review panel and the NIH VIR-B study section. He is currently a member of the American Cancer Society's Council on Extramural Grants, a member of the governing board for the ICE-HBV Consortium, seeking to guide evidence-based efforts for a HBV cure, and chairs the Scientific Advisory Council for the annual International HBV Molecular Biology meeting. Our second speaker today is Dr. Chari Cohen. She is the Senior Vice President of the Hepatitis B Foundation in Doylestown, Pennsylvania. For the past 20 years, she has planned, implemented, and evaluated community-based programs and research projects focusing on hepatitis B and liver cancer. Her research focuses on reducing hepatitis B-related health disparities, including developing culturally competent models for improving healthcare access. Dr. Cohen received her MPH from Temple University and her DRPH in Community Health and Prevention from Drexel University Dornsife School of Public Health. Our third speaker for today is Dr. Jake Liang. Dr. Liang is currently the Chief of Liver Disease Branch and Deputy Director of Translational Research at NIDDK at the National Institutes of Health. He graduated from Harvard College and received his MD degree from Harvard Medical School. He completed his internal medicine residency at New York University Bellevue Hospital Center and a fellowship in gastroenterology and hepatology at Massachusetts General Hospital and Harvard Medical School, where he was an assistant professor of medicine from 1990 to 1996. Dr. Liang is an internationally renowned and dominant thought leader in the field of viral hepatitis and liver diseases, with diverse scientific expertise and editorial experience in virology, immunology, cell biology, therapeutics, regenerative medicine, and genetics. He has published over 300 scientific papers, with many of them in high-impact journals, including the New England Journal of Medicine, among others. Our fourth speaker today is Dr. Sue Wang. She is the medical director for Chinese Medical Program at St. Barnabas Medical Center and the current president of the World Hepatitis Alliance. She is board certified in internal medicine and pediatrics and primarily specializes in adult primary care. She received her medical degree from the University of Miami and her master's in public health from Johns Hopkins University. She serves on the board of directors for the Hepatitis B Foundation, acts as an executive board member for the World Hepatitis Alliance, an international partner with the World Health Organization, and she also co-chairs the New Jersey Hepatitis B Coalition. At this time, I would like to welcome our first speaker, Dr. Tavis, to share a bit about HBV cure science and research progress.
Well, hello, everybody. Need my slides here in a bit. I'm sure they'll pop up in a moment. I have two tasks today. The first is to describe a little bit of what HBV cure science is. And then my second task after that will be to give a couple of highlights as to the progress that has been made over the last year in HBV cure discovery. And slides, please. There they go. Okay, so to get started here, HBV cure, why do we need it? The reason for that is the current drugs, although we have some good drugs, they have some big limitations. The biggest of them, I think, is probably that they rarely cure patients and they don't stop disease progression in everybody. So even if you're taking the drugs for an extended period of time, the virus is still there, and that's a problem. Second, the dominant class of drugs, which is the nucleoside analogs, they need to be taken more or less for life. And taking a drug for that long may have unanticipated side effects, and certainly it's not an enjoyable aspect for the patients. So the other problem with them is that we really have only 2 flavors of drugs for HBV, although there's something like 7 of them that are approved. All of the nucleotide analogs work in the same way against the same viral target. They really are variations on the same type of drug. And then, of course, the other major class of approved drugs is the interferon alpha derivatives, and they all stimulate the same set of cellular immune responses against it. So they really have only 2 mechanisms of action against the virus, and that's clearly not enough. Next slide, please. So what's the biggest obstacle to curing HBV? Why is it so darn hard? Well, that has to do with a molecule called the cccDNA, for covalently closed circular DNA. That is the central molecule in HBV replication because it's the template for all of the HBV viral DNAs, and it's the master copy of the viral genome in the cell. So it's basically the core element of the virus within the cell. And the big problem with this one is it appears to be very long-lived in liver cells. If it's really long-lived, you don't have a lot of handles to dig it back out of those cells. So that's the problem that we're facing right now. So next slide, please. So how do we get rid of the cccDNA? Well, the big problem with that is nobody knows. So there's an enormous amount of research going into that right now. But we've got a couple of advantages. And the biggest one I've italicized there is that clearance of an acute infection gets rid of the vast majority of the cccDNA perfectly safely in human beings. And the important part about this is that the immune system can do it. That means that we're trying to develop a cure for HBV, but we know it's possible to be done. And it's much easier to get something done if you know it's possible than if you're struggling with not even knowing whether or not it is conceivable. So a couple of other quirks on the cccDNA is, first off, it's not always completely eliminated, even during successful resolution of an acute infection. However, our immune systems come to our rescue again, and in almost everybody, the immune system can keep any little teeny bit of residual cccDNA that remains in the patients under control for the rest of the life of the patient. So again, the immune system is our big friend here. Next slide. So what do we mean when we talk about HBV cure? So as many of you have probably heard, the goal of cure research is to develop a functional cure. And this contrasts to something called a complete or sterilizing cure that completely eliminates the cccDNA, where a functional cure does not necessarily do that. So a functional cure is defined as no detectable cccDNA in cells or HBV DNA in the blood, no disease progression, and immune control of any residual cccDNA in the body. Remember that no detectable cccDNA doesn't mean that it isn't there. There may be trace levels around, but we need the immune system to go in there and keep a lid on that. So that is what it is. The clinical definition, which is what's used to determine whether or not somebody's achieved a functional cure, is undetectable HBV DNA and HBV surface antigen in serum 6 months after cessation of treatment. And of course, this is defined that way because that's what we can measure. All right, next slide, please. So what will a cure therapy look like in the long run when we're all done and we've got good cures available? It'll be a combination therapy, and that will be needed because HBV is very genetically diverse. There's lots of genotypes around the world, and there's a very variable disease course in each individual patient. And together, this means that no one drug is likely to be able to cure everyone. The other issue is that the CCC's durability means that we're going to have to hit it from multiple angles at the same time. This thing is going to need the 1, 2, 3 punch to get rid of it. So the other thing is, and this is my opinion here, is that cure therapy is likely to be long, maybe a year, I don't know, nobody knows, but it's likely to be a relatively long-term treatment course and need to be use exceptionally safe drugs. So these are things that we know or speculate about. Next slide, please. So what type of drug discovery work is going on? The cure work falls into basically 3 categories. The first of these is direct-acting drugs that target HBV itself. For example, that is what my lab is involved in. There are also host-targeting drugs that cause a patient's cells to block HBV replication by hitting an actual patient or a human target. And then there's immune-stimulating drugs that train the patient's immune system to attack HBV. This work is being done all over the world by an enormous workforce of very dedicated scientists, being done in universities, in academic and in government laboratories, in biotech companies, and enormous pharmaceutical companies. So basically, everybody who can work on this is attacking it as hard as they can. Next slide, please. So this tells you just a little bit about how we're going about doing it. I'm not going to go through this slide. It's way too busy. If one of my students made a slide like this, I'd yell at them for it. But in any case, the point here is its actual complexity. We're hitting every single vulnerability that HBV has that we know of, and we're also actively looking for more vulnerabilities. So the point here is just to say that there is a full court press going on worldwide to try and cure HBV. Next slide, please. So I'm now shifting gears. This is now a research update for 2020. This is highly personal. If you ask 3 hepatoneurologists what they think was the most important over the year, you're likely to get 5 different opinions. So this is impacted by my own personal opinion, but I do feel this is quite important. Next slide, please. One of the ones that I think the thing that I found most exciting over this entire year was that we now have a better idea of what the half-life is of the cccDNA. Half-life is the word that describes how fast the cccDNA breaks down. And this is important because for a long time, people have thought that the cccDNA had an extraordinarily long half-life. Some people thought it is approaching infinity. But that's in a long apparent half-life because all of the previous data assumed that the therapy that we currently have completely stops replication. But those of us who have studied replication for 20 plus years, like I have, know that that's not the case. And so a group took a very novel approach to studying the half-life of cccDNA, and they looked at evolution and resolution of drug resistance cccDNA molecules. And they found a much shorter half-life than was expected, and it wasn't dependent upon assumptions related to the efficacy of the drugs that the patients may have been on. And so these studies revealed a half-life of 11 to 22 weeks among the 10 patients that were studied. This is much shorter than what used to be believed. And this is extremely important because the shorter the half-life indicates that the virus is turning over in the liver. That means there's more handles available to us to attack it, and we can get rid of it easier and quicker. So this, I think, is some extremely important information. Next slide, please. Another cool one that came out is that there is a direct, explicit demonstration showing that even under really good nucleoside analog treatment, that's like tenofovir and entecavir, they can reduce the HBV replication well below the limit of detection or quantification. But of course, the lower limit of detection does not necessarily mean not there. It just means our assays aren't sensitive enough to detect it. And so these researchers took serum from patients with unquantifiable DNA levels. They're very, very, very low, and used those to infect a type of mice called chimeric mice that actually have human liver cells replacing the mouse liver cells, and they're infectable by HBV. And they used the serum from patients with unquantifiable DNA in their serum to infect these mice. And as you can see on the slide on the right hand there, the image on the right hand side, the graph, you can see that in all of the infected mice using all of the serum, HBV started replicating right away. So that tells us not only is there a trace amount of HBV DNA still around, but also that it's functional viruses. It's in functional viruses. So this tells us that replication is not completely shut off, and we have to do that better. Next slide, please. So now I'm going to go over a couple of classes of developmental HBV drugs for which there's been good advances over the last year. The first of these is the capsid assembly modifiers. These are compounds that interfere with capsid formation. Capsid formation is the protein shell that surrounds the viral DNA in the virus particle. So these molecules are in phase 1 clinical trials. There's 5 of those, or have progressed into phase 2 clinical trials. There's 4 of those. These have very promising antiviral efficacy. There's no doubt that this is going to be a good class of drugs. Just so for an example, 32% of the patients in a small study treated with one of Johnson & Johnson's experimental drugs had undetectable DNA at day 29. So it's clear that these drugs are working quite well in the clinical trials. Now, some of them have already failed due to toxicity present in human beings, but the leading compounds appear to be very well tolerated, at least as far as we know at this point, although there's a lot of work to be done with them yet, but they do appear to be well tolerated. Another potential weakness of these guys is that HBV develops resistance to some classes of them relatively easily, but that's likely to be manageable because all of these compounds are going to be used in combinations with multiple other antiviral drugs, and using drugs in combinations helps suppress resistance. So I am quite bullish or optimistic about the capsid inhibitors. Next slide, please. Another one that I think is really cool is a drug that was originally called Merculedex B and then went through the name Hepcludex, and now is, oh God, I can't even pronounce this one, bulevirtide, I think. And this is an injectable drug that stops HBV from entering cells. So it's a class of drugs called an entry inhibitor. This drug was just conditionally approved in Europe against hepatitis delta virus. Now you're all saying, hey, this is an hepatitis B virus webinar. Well, hepatitis delta virus is important because it's always found in the presence of hepatitis B virus, and also it enters the cells using the same mechanism that HBV does. So if it blocks hepatitis delta virus, we know it's going to block hepatitis B virus. And from lab experiments, we know that very clearly. And it's also in phase 2 clinical studies versus HBV, where it's very safe and effective so far. So I'm also very excited about Merculedex B, and I really think that it will be approved in the relatively near future as soon as the clinical trials are done. Next slide, please. The last major thing that I want to talk about here is some interesting information that's come out about small interfering RNAs or siRNAs. And it's found that at least some of them can durably suppress HBV long after the drug is finally given. Now, an siRNA is a little teeny piece of RNA. It's kind of related to DNA. And what it does is it shuts down production of the viral genetic information and consequently blocks viral replication. And so that stops production of the viral proteins. there's no doubt that this can work against HBV, and this has been known for a very long time. Now, there are a number of different drugs. There's one in phase 1. There's one in a combined phase 1, excuse me, there are 2 of them in a combined phase 1/2 study, and there's a couple of them, 1 or 2 of them out in phase 2 studies right now. One of the results that came out recently that I think is most interesting is that about 15 out of 38 patients who responded well to, again, another Johnson & Johnson product had durable post-treatment suppression of the viral surface antigen in serum at 1 year, with an average suppression of a 2 log 10, that's a 100-fold suppression at 1 year. And the cool thing about that is at 1 year, there's none of the drug left in these patients. That would have cleared by this point. And so the durability implies that there may have been some reactivation of anti-HBV immune responses, so the immune system may actually be helping here. So I'm also very optimistic about the siRNAs. I will be quite surprised if, when the clinical trials are completed, that none of these achieve approval and become relatively frequently used in cure therapies. Next slide, please. Okay, and yeah, there's one more. Sorry about that. I can't count today. Then there's the functional cure by the nucleic acid polymers. This is through some studies being done by the Replicor company. Nucleic acid polymers are polymers or strings of molecules all hooked together that block secretion of the HBV surface antigen from cells. There's a phase 2 study that has just been completed with 40 HBV antigen negative patients. There was a triple combination studies using NAPS plus tenofovir plus pegylated interferon alpha. So this is a triple therapy with a whole bunch of things being thrown at the virus. The cool thing about this, and I do think it is truly cool, is that 39% of the patients in this small study who managed to complete the 2-year trial achieved a true functional cure, as near as we can tell. These patients seem to have achieved a functional cure. And so that is much, much higher than the rates of, 5%-ish of what all the other therapies, the currently approved therapies, are getting. So if this holds in later studies, this could be a huge advance. Now, some additional information about this. Hepatic flares occurred in all patients with good responses. They were manageable, but they do need to be followed medically. And this requires weekly injection of the interferon and NAPS. So it's not necessarily going to be the easiest therapy to take. But in any case, this is the best that science has been able to do so far. And I'm really quite excited about this one too. Next slide, please. So the take-home message from this portion of the webinar is that our goal is to develop a functional cure so that we can help as many patients as possible. A functional cure is going to require combinations of drugs that work in multiple different ways. We have continued to improve our understanding of how HBV persists in the body over the last year, and that's being very useful in guiding development of the cures. There's a whole raft of promising drug candidates that are in the process of being developed, excuse me, and some of them are looking really good right now, and functional cure rates of up to about 40% have been seen in a small phase 2 study. So the big take-home message here is there is hope. I do think, and I truly believe, that a functional cure, and a widely applicable one, is going to be coming, and going to be coming in a reasonable time frame. So I'm excited. Thank you very much.
Great. Thank you so much, Dr. Tavis. Now we'll turn it over to Dr. Chari Cohen to speak about HBV cure community perspective. Welcome, Dr. Cohen.
Thanks so much, Catherine, and thank you, Dr. Tavis, for talking to us about your thoughts in the latest in cure research, and frankly, for giving us hope. I think it's something we could all use a little bit more of. Catherine, next slide, please. Thank you. So I'm going to talk a little bit from the community perspective about what people with hepatitis B document that they want and they need in terms of treatment and potential cure or functional cure for the future. So this is a project that we started about 12 months ago with Hepatitis B Foundation to try and document patient wants and needs. And one of the overarching goals was to document the impact of living with hepatitis B. So for people who are chronically infected, what's the impact on their physical, emotional, professional, and social lives? We wanted to ask questions about experiences with stigma and discrimination as well, and how that impacts their lives. And then we wanted to talk to people living with hepatitis B about what they prioritize in terms of treatment. What are their current treatment experiences and challenges, and what are their thoughts for the future? What would they like to see in an ideal treatment? And hopefully we can use the data that we're documenting to improve clinical trial participation. As we know, there are new clinical trials coming down the pike, and we want to make sure that people living with hepatitis B participate in these clinical trials so that we can get towards that functional cure. And we also want to ensure an improved diversity of people who are participating in clinical trials as well. And we were hoping that if we could get more information about patient-reported outcomes, that we could include that in some future, potentially share it with people who are developing clinical trials and who are developing treatments, and help include patient-reported outcomes into treatment regimens for the future. Next slide, please. So this is what we've done for the past 12 months. We had a mixed methodology. So first we hosted our externally led patient-focused drug development meeting, lots of words there, in June of 2020. And that was a qualitative way to document the needs of the public. And I'm going to be talking a little bit about that today, about some of the results from that. We also conducted 25 in-depth interviews with people who have chronic hepatitis B throughout the U.S. These were 60 to 90-minute interviews that we did over the phone, and we then we transcribed the data and are in the process of analyzing, actually almost done analyzing those data. We completed data mining and a qualitative analysis of about 10,000 emails coming from people living with hepatitis B around the world through our consult line. And then finally, we conducted an online survey, and I'm also going to share a little bit of results from that today. And the survey was both focused on people living with hepatitis B in the US and internationally. We were going to be hosting focus groups in 2020, but because of the coronavirus pandemic, those got pushed off until next year. And we might wind up doing those remotely instead of in person. Next slide, please. I'm going to start with a couple of findings from the interviews that we did, the telephone interviews and the PFDD, and these are qualitative findings. So starting with the impacts of chronic hepatitis B, there does appear to be significant physical and emotional impact for people living with hepatitis B, and it seems to reduce quality of life and reduce enjoyment of daily life as well. People mention quite often fatigue, feeling fatigue. So fatigue was the number one in the survey on the PFDD and the interviews. Fatigue was the number one physical symptom that was reported and pretty widely reported as well. But also things that we don't talk about as much, like shame, people feeling shame and stigma, people feeling isolation because of their hepatitis B, feeling that they have to keep away and feeling they're self-isolated, but also feeling that other people stay away from them, that other people isolate from them. And then the number one emotional impact and fear was fear of liver cancer, fear of dying prematurely because of their hepatitis B. In addition, we also found that people living with chronic hepatitis B had impacts on both their familial and their social relationships, as well as education and jobs and careers. And the impact seems to differ a little bit internationally versus US. I think because of some of the policies and protections for people around the world, or lack thereof. But we did see that people living with hepatitis B, that throughout the lifespan, it seems to impact their family and social relationships, including marriage and children. We also found that there seem to be many current treatment challenges for people both in the US and outside the US, including cost of medication, access to medication, and also taking a daily pill for many years, which is something that, we haven't necessarily thought about. I think a lot of times from our perspective, it seems pretty simple. Oh, you take one pill every day. But honestly, it's a burden for a lot of people, and qualitatively people stated that number one, not only is it a burden because they forget often to take it, but people feel like it is a constant daily reminder. And a lot of folks talked about that on the PFDD and in the interviews as well. And it's not something that people prefer to do. And that, of course, there's a strong desire for loss of surface antigen and risk reduction for liver cancer, ideally with a finite treatment, a treatment that would not need to be taken for many years. Next slide, please. In terms of future treatment, from both the interviews and the PFDD, what we heard was that there was a preference for oral treatment, but that injectables would be considered acceptable by many people. Additionally, mild and limited symptoms were considered to be acceptable by most people. However, there were some caveats with some of this, and a lot of it did lead back to interest in clinical trial participation. So, for example, people were concerned that there would be clear safety information before they would consider participating in a clinical trial, and they want assurance that there would not be long-lasting damage to their liver, but also to other organs in a potential new treatment. Additionally, people were looking for treatments and clinical trial opportunities that would be less disruptive on their daily life. So, potentially thinking about how long they would have to travel for treatment or to a clinical trial site, what the side effects would be, and the amount of impact of the side effects on people's lives, and timing as well. So, for example, how long would they have to be in a clinical trial, 6 months, 12 months, or longer, and how often they had to travel to the site for testing. Additionally, can you just go back one quick? Sorry. Additionally, we saw that both people who had young children who participated in the interviews in PFDD and people who were older seemed to be more hesitant about participating in clinical trials. And some of what we were hearing was that, they were feeling comfortable, especially people who were older were feeling comfortable with the treatments they were taking and were less likely to want to rock the boat, especially if they had comorbidities, and were just a little bit more hesitant about potential participation in clinical trials. And then finally, it seemed as though a success rate of between 50 and 70% would be needed for most people to choose to participate in a clinical trial. Next slide. I'm going to switch over. So these are very preliminary results. We just started mining through the survey data about a month ago, and we're still working on our correlation studies, but this is preliminarily. We had a little over 1,700 responses for the online survey. The age ranged from 19 to 75. I will say about 50% of survey respondents were between the ages of 18 and 45, which I think is great because those are the people, we often see impact midlife and really do want to get the thoughts, especially people who might be more inclined to participate in clinical trials in the future. 83% of our respondents lived outside the US and represented 99 countries. And the most represented countries were Nigeria, US, Ghana, India, and the Philippines. Next slide. So this is data from one question that we asked. We asked people to rank the importance of different future treatment outcomes. And overall, so this slide represents combined results from the US and international respondents. So overwhelmingly, loss of surface antigen was considered to be the most important future treatment outcome, followed by being able to stop medication after 6 or 12 months. Third down the line was having a decreased risk of liver cancer, followed by having improved quality of life, and then rounding it out at the end with loss of cccDNA and undetectable HBV DNA. So then if you head on to the next slide, what we did was actually broke this down then and looked at it by where people lived. So you can see while loss of surface antigen overall was considered to be the most important treatment outcome, if you compare the results in US versus international respondents, it actually differed. So looking at the top 3 for US respondents, the number one most important outcome was considered to be decreased risk of liver cancer. Internationally, however, it stayed as loss of surface antigen. And so, these are results that we can follow up with some qualitative studies and try and figure out a little bit more in depth what's happening here. I suspect that a lot of this is because surface antigen is where the discrimination pops up around the world, right? People who test positive for surface antigen, they're the ones who can't go to university or can't get jobs. It's that surface antigen that are holding people back from life. And so I'm not surprised that loss of surface antigen would be considered to be most important among respondents internationally. Same thing with the number 2. You see loss of surface antigen pops down to number 2 in the US. Number 2 priority outside the US is being able to stop medication after 6 to 12 months. And again, I have, I suspect, and this is just my personal opinion, but I suspect we would find that this has something to do with access. People around the world are not necessarily able to access medication. It's difficult, it's expensive. And so that might have something to do with why we see the differences here between US and international. Next slide, please. We did ask people what acceptable side effects they would consider when trying to think about joining a clinical trial for a potential functional cure. So it looks like the majority of people, a little over 50%, were willing to accept something like mild headache or stomach upset for a few days. And then it kind of dropped significantly. So some people were willing to accept more longer, mild or more severe, I guess, than just mild headache, but mild to moderate flu-like symptoms for a few days. And also there were a small percentage of people willing to risk an ALT flare, and also mild to moderate fatigue for several days. But you can see a pretty big drop off there. And I think side effects are a big concern for people when they're considering whether or not to participate in a clinical trial. Next slide. We asked people what their willingness to join a clinical trial would be for a specific chance of functional cure. And so you can see here, the blue bars are international respondents and the orange bars are the US respondents. So for a 25% chance of a functional cure, 39% of international respondents said that they would consider that they would be willing to join a trial versus only 16% in the US. For a 60% chance of a functional cure, 62% of international respondents said they would be willing to join versus 43% in the US. And then finally, for a 90% chance of a functional cure, 85% of international respondents said they'd be willing to join a trial versus 74% in the US. Next slide. So this is just kind of a sneak peek of the things that we found from our PFDD and interviews and initial analysis of the survey. We will be releasing the PFDD report, the report from the patient-focused drug development meeting. It will be released sometime in October, probably within the next couple of weeks. It's just going through final approval process. So hopefully all the data that you saw here will actually be released very soon. The rest of the data we will be sharing widely as we analyze and get more familiar with the results. But I just wanted to end my part today talking a little bit about what happened to the cure. And this is something I think is important to talk about because, with the Hepatitis B Foundation, we take about 10,000 consults a year from people living with Hep B, and this is one of the most common questions. Where's the cure? Why don't we have a cure? 10 years ago, you said there would be a cure. It's 10 years later. What happened to it? And I think we need to do a better job communicating around the world to people about the process of drug development, what it takes to develop a cure, and what progress we've made. I think webinars like this are a really good start. And if we could find more ways to share the information, I think it would be really helpful. I'm hoping this can be some kind of a call to action to help prioritize hepatitis B. I think if we can inform and empower people who are impacted by Hep B and tell them how much progress we've made and what we need to get over that line. There's a lot of hope, but we still need more prioritization for Hep B. We need more funding for research. I think if people knew that, we could help empower them to advocate for these things so that it would bring us over the edge. Again, I just think it's really important to get people ready now, because if we do see a functional cure within the next few years, getting people tested and into care and improving capacity around the world to get people the drug will be really important. And with that, I'm going to sign off. Thank you so much for letting me chat today.
Thank you so much, Dr. Cohen, for sharing a bit about the patient perspective from different projects ongoing at the Hepatitis B Foundation. It's always really important to keep that in mind as we talk about cure and the path to a cure for hepatitis B. At this time, I would like to turn it over to Dr. Liang, who will be talking about NIH and ICE-HBV initiatives towards a hepatitis B cure. So at this time, Dr. Liang, I'll turn it over to you.
Okay, can everybody hear me?
Yes, we can hear you.
Yes. Okay, I guess maybe my webcam is not working, so you don't see me on the screen, which is fine. I may have a bad hair day, as people call it, so if I have any hair at all. So I just want to kind of, again, thank the Hepatitis B Foundation for hosting this webinar, and certainly with Dr. John Tavis giving an introduction on the science of HBV cure makes my job much easier in terms of talking about the various initiatives that I'm going to share with you that this particular organization I've been associated with, ICE-HBV, as well as NIH, federal government, have begun regarding an effort to cure hepatitis B virus. So the ICE-HBV is a, can I advance this or, okay. It's an international coalition to eliminate hepatitis B organization that was formed in 2016 and aims to establish an international collaboration to facilitate the discovery of a safe, effective, affordable, and scalable cure to benefit all people living with chronic hepatitis B, including children and people living with HCV, HDV, and HIV co-infections. And so the effort is really not just on hepatitis B, but also in many other infectious diseases that may coexist with hepatitis B. Next slide, please. So the ICE-HBV was formed back in 2016, as I said, and it sort of began sort of a whole dialogue about a global scientific strategy towards a cure of chronic hepatitis B virus infection. The organization was basically started by a group of us, and sort of we are currently the governing board, and the member of which, including Dr. John Tavis and Joan Block, you're going to hear from her later. And we are advised by a group of senior, well-known, esteemed scientists and clinicians. And we work closely together with various different stakeholder consulting groups, as well as scientific work groups. And at this point, there are 3 scientific working groups, one focusing on virology, the other one on immunology, and the third one on innovative tools that can expedite the discovery of curative therapy for hepatitis B. Obviously, integrated within these the various groups, the clinical science is certainly very critical in understanding how we can achieve curative therapy, although we don't really have a separate clinical science work group, but we certainly work very closely with many clinical consortiums and trials, and even pharmaceutical companies trying to bring about a cure for hepatitis B virus. Next slide, please. There are really several actions that the ICE-HBV have articulated in terms of strategy, and they are actually very much appropriate for the acronym of our organization. So I is to increase funding for individual and collaborative cure-related research projects by governmental and private funding agencies and philanthropic organizations. So the HBV cure research investment strategy should be prioritized in national plans in a very global way, no matter which region you are, because as we all know, hepatitis B is a global disease. So ICE-HBV has played a very active role in contributing to this inclusion of hepatitis B virus in the G-Finder report, which is a very influential yearly report on neglected diseases on the global scale. So to be able to highlight hepatitis B as a neglected disease certainly should bring much attention to what we're trying to do. And also, as I mentioned, the hepatitis B elimination should be included in the global health program. For instance, with this, again, another very influential organization, the Global Fund, Replenish, has a meeting in 2019, and in this meeting, the hepatitis B was also featured in some side events, and certainly has broadened the appeal and bring much awareness about hepatitis B virus. And also WHO, as many of you know, obviously, World Health Organizations has certainly been on aligned with the whole concept of the viral hepatitis eliminations is very important in terms of the global universal health coverage, and that was highlighted in March 2020 in some of their meetings. So in a way, I think we are able to bring hepatitis B virus to the radar screen of many of these influential international organizations, which in the past has not been so for hepatitis B virus. Next slide, please. So the second action is the C, is to concentrate on discovery of therapeutic strategy that will permanently reduce the number of productively infected cells and or permanently silence the cccDNA in these cells, and that will, and also the strategy will stimulate HBV specific T cells and the production of antibody that will prevent viral spread to uninfected cells, basically mimic spontaneous resolution of HBV infection, and in sort of maybe in more term, it's called a functional cure that you heard from both Tavis, Dr. Tavis and Cohen. And so the couple of specific actions that have taken place over the last year, one is there will be a HBV cure serum biomarker workshop that will take place next week. And this is really to address the very different markers that we've used and how effective they are being used to expedite the development of curative therapy. And there's also an ongoing cccDNA working group, as you heard from Dr. Tavis, that the cccDNA is really the master template of hepatitis B virus in terms of transcription and replication. So it's a very critical target that we need to try to eliminate. And then also there will be a HBV cure webinar workshop at the conference on liver diseases in Africa. That's 2020. As certainly many of you know, that hepatitis B virus is endemic in many parts of Africa. So it's very important for us to pay particular attention to this particular region, which has been neglected in various different global efforts in terms of eliminating infectious diseases. Next slide, please. So the last action is to establish repository of standardized HBV reagents and protocols, and to facilitate access to all researchers across the world and support the development of a new animal model. So again, this is really trying to develop tools that we can facilitate the discovery of curative therapy for hepatitis B virus. To that effect, can I have the next slide, please? There is already a so-called ICE-HBV open access protocol database that is spearheaded by Hightower and some of the other scientists in the HBV field. And many protocols has been deposited in virology in 2019, and they're trying to do the same thing with various different methodology and protocol in the area of immunology, which I'll talk a little bit about in the slide later. And then there's also, well, let's go back to the previous slide. There was also an international workshop that was part of the International HBV Symposium back in 2019 in Australia, in which there was a very active discussion about how we can develop a very effective in vivo models that can be used for the development of HBV curative therapy. And then last is part of the NIH effort, which I'll tell you a little bit about later too as well, is that as many of you know, that there are a lot of reagents that have been developed by various different pharmaceutical companies, private laboratory, academic institutions. So it would be very critical to make a repository that can be shared with the rest of the world in order to spur the discovery of hepatitis B virus. So the next slide. Next slide, please. So just again, this is the sort of repository I was just referring to, and it's underway. It's basically sponsored by the National Institute of Allergy and Infectious Disease, it's part of NIH. In this particular repository, many of these very useful reagents, I don't need to go through them, such as peptide library, antibodies, viral constructs, and cell lines, and even a serum repository and liver sample from HBV patients, hopefully will be put in the central repository for everybody to have access to and in order to use. I think this would be a very important effort that is underway. Next one. And then also, I just mentioned about in companion with actually reagent, there will be various different protocols that have been developed by many scientists in the world that will complement the reagent repository. And initially, this repository is focusing on virological protocols, and some of them are listed there, such as cell culture, how to grow the virus, how to assay them, what kind of assay do we have, and in vitro cell culture systems. So that's sort of actually a very long line. So on the right-hand side, you can see that there's a sort of the web interface of very easily navigated website that you can find whatever protocols that you need. And so this is something that's still a moving target and also a living document, because we hope to expand on this particular websites. And as I mentioned, the next effort is really to include very different immunological protocols and methodology in this website. Next slide, please. So what are the next steps that ICE-HBV have undertaken? It went a little too fast. I guess I don't have control over this, but anyway, there will be a ICE-HBV sponsor think tank on HBV cure in the next EASL meeting in Amsterdam, April 2021. This took place a year ago in the EASL meeting in I think London, similar type of format. So that was very well attended and certainly raised a lot of awareness. And there will be a public forum as well in the next international HBV meeting in Toronto, and it was supposed to be this year, but obviously was postponed to next September 2021. And we held similar event like this in the last HBV meeting in Melbourne, Australia. It certainly had gained a lot of traction, a lot of publicity, at least in Australia. So I think that's hopefully in a different continent we can do the same with raising awareness. And then there's also a very important area. I mean, there are vast number of countries that so-called resource limited region that may not be able to manage HBV as effectively as in Europe or America. So I think that's a very important point. And so ICE-HBV, led by Daryl Lau from Harvard, then they are kind of trying to organize, looking at how can we address some of the resource limited setting once we have better therapies. And I think that's very important. I think that will be very, very important topic, especially as ICE-HBV is a global organization. And these are some of the photographs on this showing you some of the meetings. Next slide. And this is just sort of a share with some of our financial supporters. Obviously, we can't run the organization without the support of various different organizations. Some of them are pharmaceutical companies, some of them are foundations, and as well as some of the non-government agencies. So I think that's just very important to have the support. And I would just have to certainly highlight the fact that none of these supporter donors have any influence and dictate any of the priority of what we do. So I think that's very well understood, and they certainly respect the boundary between what we do versus the financial support they have provided. And lastly, let me just want to share you briefly about the U.S. government's effort in terms of HBV cure. Again, ICE-HBV as well as HBV Foundation have played a very active role working with National Institute of Health to come up with very different strategy to cure HBV. And so, which is a wonderful opportunity to be able to combine a lot of these sectors to come up with the best way to approach curative therapy. And from the NIH point of view, actually, not just NIH, this involved the whole Department of Human Health Service, including CDC, FDA, and other federal agencies. This began with this a publication in 2011, which basically put together this U.S. National Viral Hepatitis Action Plan, was in a document that's been widely distributed. The document is title is Combating the Silent Epidemic of Viral Hepatitis, and lay out various different action plans for the prevention, care, and treatment of viral hepatitis. Of this action plan, HBV cure and prevention is certainly a major focus. And then subsequent to the initial publication of this document, there have been several iterations or renewed efforts and refocus from 2017 to 2020, which is coming to an end. There will be a revised version or updated version of this action plan from 2021 to 2025, which should come out beginning of the next year. In addition to this sort of a global viral hepatitis action plan, NIH actually has been very active, involved in specifically focusing on the curative therapy for hepatitis B, and again, as a partner of various different NIH institutes, that there was a strategic plan for trans NIH research to cure hepatitis B that was published late last year, and that certainly have received a lot of interest and attention. Again, Hepatitis B Foundation and ICE-HBV have played an active role in terms of encouraging these kind of federal strategic plan. And the result of this certainly have, we've done very kind of a concrete, very specific and concrete way. If you look at the NIH funding back in 2010, there was only about $15 million in terms of federal funding on HBV-related research, but it's gone up to $70 million in 2020. So it's a certainly significant increase. Most of these funding are on research grants, and some of them to stimulate small business contract, antiviral testing service, and even clinical trials. And we all understand that this even $70 million is not enough to have curative therapy, but this is certainly a trend to the right direction. So hopefully this will continue to go up. And lastly, I just want to again reiterate sort of this repository HBV reagent, which is sponsored by NIAID, and that will be in place hopefully by the end of the year, that people should be able to access to, and that will be a really welcome repository that all the scientists would be able to share and take advantage of it. So to the next slide, I really just want to end my talk with a sort of an optimistic view of the cure for hepatitis B virus. Again, I think I don't really want to commit myself to how many years we're going to have curative therapy, because I don't want to be held accountable for it a few years later. But I certainly think the prospect is wonderful. And you heard from Dr. Tavis about many of the approaches, again, not just pharmaceutical company, but academic institutions, federal government. We all are very focused and interested in developing curative therapy. So I think the future is really very bright to have a curative therapy. Obviously, the recent COVID pandemic has put a lot of these other things on hold in terms of sort of put them on the back burner, particularly some of the important diseases like hepatitis B. But we all know that COVID-19 is not going to last forever, and hopefully in a couple of years we'll have an effective vaccine and certainly therapy, and so maybe they will become more like a flu, and then we can certainly we should continue to focus on hepatitis B, because hepatitis B is not going away. So that's going to take a really conservative effort among all of us in order to come up with a curative therapy. So I think the future is bright. I really look forward to many more opportunity to interact with all of you and other colleagues and friends to come up with curative therapy in the next 5 to 10 years. Maybe that's the horizon I can give you. With that, thank you very much for your attention. I'll stop.
Thank you so much. At this time, we'll turn it over to our final speaker, Dr. Sue Wang, who's the president of the World Hepatitis Alliance. Dr. Wang, I'll turn it over to you.
Great, thank you, Catherine. Of an airplane I was looking at, I was thinking, wow, it feels so foreign looking at a picture of an airplane, because we haven't traveled in so long. At this point, I think we would have seen each other at so many meetings by this year, and so it's good to have opportunities like this to connect even virtually and make sure that we're moving things forward with Hep B cure and elimination. So glad to be here. I'll be talking about patient stigma and how lived experience can drive change. Next slide. So I want to start off with some voices of people living with Hep B around the world. These are quotes taken from messages that Hep B Foundation has heard from patients directly through that patient consult program, which Chari talked about. And I want to thank Maureen for the slide. So this person says, I am embarrassed and paranoid about sharing my status with friends and family, even healthcare professionals. During my first appointment, my GI doctor said to me, you should have known better, when I shared how I contracted Hep B. So there's that stigma even from healthcare professionals. Africa looks towards the Western world in finding permanent cure for hepatitis virus. As of now, many will continue to die infected and get isolated due to lack of timely medical interventions and stigma due to economy. Honestly, there's too much discrimination in my own country about this Hep B. I feel down about this. All my dreams seem to be eliminated by this disease. I just got diagnosed of chronic hepatitis B, and to be honest, I feel like committing suicide. I'm just 23 and I've only had one partner my whole life. I'm positive with Hep B since a year now. Oops. And I really need help. I have tried different types of drugs, but it's still the same. Please help me. I cannot even afford for all the tests needed. Please help me. It's like I will die soon, please. So you hear a lot of the desperation and concern from patients, and as a physician and a patient, often in certain forums like this, many years, I never heard any of this kind of desperation because they don't necessarily share it in the exam room. But that's why I think it's important for us to be really aware of it, because it does drive, the urgency of cure. So next slide. Next slide. Catherine, I don't see the next slide, but I don't know if that's because is the screen frozen?
It might be. Right now I'm seeing the World Hepatitis Alliance Find the Missing Million page.
Is it just my end, or can you guys, sorry? Oh, there we go, okay. It's estimated that there are 325 million people infected with Hep B globally, and upwards of 290 are undiagnosed. We believe that a huge role is prevention, but because they're not even diagnosed, they cannot partake. So while we talk often about how we don't have a lot of resources in Hep B work and research and funding, we do have the power of the people, meaning that we have millions of people who are living with it, who could really, really help drive, find the movement, and that will drive change. Once patients are diagnosed, they can demand, they can drive their innovation and ultimately save lives. So that's kind of the big picture. Next. Is it lagging, Catherine?
Yeah, I think your connection might be a bit off.
All right, there we go. So the work of the, oh, okay. So you're moving it, but I just can't see it. Is that what's happening? Because I could just keep talking
Sure, yeah.
rather than wait.
You.
Okay. So the Alliance, the work of the Alliance really seeks to put patients first and front and center in the movement. We believe that people living with Hep B and the affected community should be at the heart of every effort. Patients can bring fundamentally important perspectives and experiences which really greatly enhance the effectiveness of strategies and programs. Next. So we conducted a survey with civil society on how stigma from hepatitis impacts people's lives. Many of these were actually discussed by Chari, but just to show you how widely it affects people. So in this survey, 72% of people reported having that self-stigma and internalized stigma. And it may not be, these are things that you wouldn't see, as a doctor, it may not be, it's not something I would see in their blood work or even on their physical exam. This is something that's internalized. And then beyond that, 53% reported social exclusion. And as you keep going down, you see that it impacts relationships, marriage, employment, teachers experiencing teacher discrimination from teachers, or if you own a business that you might lose customers. So there are a lot of ramifications of living with the disease, which are not necessarily clinical. All right, next. So, and this is something that even affects healthcare workers who are living with hepatitis B. And this is something that Hep B Foundation was really involved with over the past number of years. In my own state of New Jersey in 2011, there were 2 medical students who lost their acceptance for school because of their Hep B diagnosis, and the Department of Justice was brought in. The following year, CDC did update their guidelines for infected healthcare workers and professionals, basically saying that there was no reason that people with Hep B could not pursue medical careers. Following that, people with hepatitis B were officially protected under the ADA, which is really a landmark move. And the DOJ, along with the Department of Ed and HHS, sent a joint letter to healthcare schools notifying them of that. But yeah, in spite of that, the Hep B Foundation still receives 20 to 30 cases a year of students or professionals facing discrimination, and the numbers may even be higher. And I feel like every time I talk to Chari, you guys are getting more and more reports of this. All right, next. All right, so all this just goes to show you how cure means so much more to patients than just the viral load or what you might see on their lab work. This diagram is from a paper that Chari and I worked on with Tavis too, who's a researcher in Hep B in Australia, also living with Hep B, and we kind of described some of these psychosocial impacts for Hep B. So a lot, primary being like internal and fear of transmission or fear of how it impacts your life. And then these other secondary and tertiary impacts, which really talk about how it impacts individuals' interactions with their community, with their healthcare providers, or even with their families, and how there's really a reverberating effect of having the disease. All right, next. So as part of this article, we also talked about why should patient perspectives be heard on the road to cure. So there are a number of reasons. One, to clarify the rationale for finding a cure, as in why does cure matter? To understand if proposed treatment interventions would be useful, as in is the cure that I'm proposing going to be practical in the real world? To avoid any unintentional harm to the affected community by potentially exacerbating stigma or some other way through these studies. And then to maintain a trusting and respectful relationship between the scientific community and affected communities. And I think Chari touched on this in terms of how we really need to communicate where we are in the cure and the search for a cure so that the community understands it. Next. So as part of that too, we think that patients should be more involved with trials as well. So Chari's talk was really eye-opening in terms of looking at what patients are looking for in cure and how cure will help people, whether it's through the loss of surface antigen, suppressed viral load, or elimination of cccDNA. And so we think involvement with clinical trials is another way for patients to really help shape so that cure is relevant. We've talked about developing some patient advisory groups for clinical trials, and so stay tuned, and we should be, that's something that's in progress. So what does that mean, patient-centric trials? So that means that patients are involved in the design and execution of clinical trials. They have input into the studies that would improve relevance to patients and encourage patient participation in trials. And how could the patient input impact a trial? Well, it could actually change the inclusion or exclusion criteria, how many visits are required by patient to participate in a visit, and make it so that outcomes are actually more patient relevant. And what they've seen in studies is that patient-centered trials are actually more likely to produce drugs that successfully launch versus standard trials. Okay, next. So, the big picture is we won't really achieve a viral hepatitis elimination without really addressing health equity. We need to make sure that whatever interventions we develop must also be reachable by the most impacted populations. With so much of the world's population and burden of Hep B being in Asia and Africa, we must realize that with the resource limitations, that can be a big barrier in getting cure to people. And so as we develop cures, we have to consider these factors if we want cures to be reachable by all who need it most. And I think this image is just really helpful for us to see that even if you have a cure, if you can't get it to people, it won't have that the desired impact. All right, next. So, and what I would say is that even with our current interventions and guidelines, there are many, many people slipping through the cracks. So even in the first world countries like the US, where the study was, this is a CDC study of more than 2,000 Hep B patients in integrated healthcare systems, it shows many gaps. So while the majority of these patients did get an ALT, 78% of them, only 37% of people were getting a viral load yearly. And 18% of them never had a Hep B viral load at all in the 3 years of the study. And in the highest risk group, the people with cirrhosis, where 100% of people with cirrhosis should be treated for Hep B, only half were actually getting antiviral treatment. And only even in this group, 11% never had a viral load done over the study period, and less than 30% were getting imaging surveillance for liver cancer. So as a physician, I can totally see this happening because of how complicated our healthcare structure is, people changing their insurances, changing doctors, high copays. I mean, we all know this if we've had to struggle with healthcare in the US or other areas where it's complicated. So we sometimes hear from people in the field, do we really need a cure for Hep B? We already have some great, safe, effective medications, and they're suppressing viral load, they're preventing cancer. Do we really need a cure? And I would say, even though we have these great interventions now, there's still so many gaps. People aren't getting tested, people are not getting the medications. Being on lifelong therapy is really not tenable for many people. And as a result of this, we are seeing liver cancer rates on the rise, both in the US and globally. Next. So this is a snapshot of all the international guidelines out there for Hep B by the American Liver Society, the Asian, European, and you see there are variations in terms of viral load cutoff, ALT, and because of this makes it very complex for physicians to follow in terms of deciding when to treat people. Next. So as a patient community and people who are advocating for patients, we really need to push for simplification of guidelines. Simplification equals access. Complex guidelines means that care will not be accessible to many people in the world, and we need it so that you don't have to see a hepatologist to get care. We've seen this happening with Hep C, simplification of guidelines. You can get cured with Hep C in just 2 to 3 visits. And we really need to move Hep B towards that as well, at least even in the care side of things. So last year, a group of us developed Hep B care management guidelines for the PCP, which I encourage people to access online, and this is just a snapshot of it. All right, next. So in closing, just a reminder, final reminder that elimination will not be achieved without affecting people who are affected by viral hepatitis. Next. And it will take a village. And this is a picture of the No Hep Village, which we had last year in Toronto at the Global Hepatitis Summit, which is a research medical summit, but as part of it, we showcased community groups that do work in hepatitis, and we had our own village in the exhibition space, and it was just really an energizing place to meet people doing good work directly with affected communities. ICE-HBV and Hep B Foundation were a big part of this. And so we're really grateful to see, more and more partnerships that have come together. We hope for more. We really need the collaborations to get us to elimination. Invite everybody to join No Hep. No Hep is what we hope is to be like the Red Ribbon campaign for HIV, but for Hep B and Hep C. We look forward to more opportunities like this to really move things forward. Thanks so much.
Thank you so much, Sue. We really appreciate you being here.
Thanks.
So I'd like to now invite all of our esteemed panelists to turn on their webcams if they're available. Just a reminder to those panelists, the webcam feature is under your microphone. If you click that, you should be able to share your webcam. For those of you who are attending today, we'll now start our question and answer session where we'll be able to ask panelists directly questions. So be sure to type questions into the question and answer box so we can facilitate discussion with you all. The first question we have is, I believe, for Dr. Tavis. This question is the cccDNA contained in those who have inactive chronic hepatitis B?
So the answer is yes. If you have hepatitis B, you have cccDNA. There are a couple of complexities on that because of integration of HBV DNA into the genomic DNA, but those get very complicated in a hurry, and it makes diagnosis or definition of them a little bit harder. But basically, if you have the chronic inactive forms of HBV, yes, you have active cccDNA in your body. It may not be very active, but it is there.
Thank you so much. So some other questions that we have coming in. So a lot was spoken about drug development. Is there a way for patients to stay informed about drug development and what's going on in the research lab related to hepatitis B, and how can they keep up with what's going on?
Did you want me to answer that one again?
Sure, we can open it up after. Yeah.
I think the best place to go to keep track of, at least the clinical drug development, is at the Hepatitis B Foundation's Drug Watch page, which they maintain on their website. In fact, I use it as a reference for knowing what's going on at the clinical level, because I work mostly at the preclinical level. As to what is going on in the research labs, preclinical, that would be a little bit harder for the layperson to access. I would recommend going to the National Library of Medicine. They have a number of tools where you can search and search for hepatitis B virus research under reviews, and then try and look for the really short ones. Those short ones tend to be more in the overview level. The really long ones tend to be really, really in-depth and, quite honestly, excruciatingly boring, even for somebody like me.
Thank you. Sherry or Sue, do you guys have anything, or Jake, do you guys have anything to add to that?
Yeah, this is Jake. Actually, I also want to point out the utility of this webinar, right? I mean, it's a really great opportunity for many people, patients, to learn about new therapy. John certainly done a fabulous job in terms of simplifying the drug development. I think these kind of venues hopefully will continue in a way that will keep the patients engaged in the whole process. And so I think that's another area that we could certainly probably do a better job in terms of spread the news.
Great. Thank you.
Yeah, so thank you for the shout out for the Drug Watch. We do update that on a daily basis. So please do go to the Hep B Foundation's Drug Watch. We have preclinical, clinical information on there. We also now have clinical trial finders on the Hep B Foundation webpage. So for hepatitis B, hepatitis D, and liver cancer. So if you were looking to see what new clinical trials might be around in your area, you could put your information and take about 60 seconds, and then you would learn if there are any new clinical trials near you. And I think social media, I've been seeing a lot lately, as companies are getting more, are seeing more results and moving towards clinical trials. We're seeing a lot of press releases, and then a lot of like ICE-HBV, Hep B Foundation, we share that through social media, and that's a great way. So I think follow organizations, and you'll get linked to some of this stuff too.
Yeah, I think one thing probably Hepatitis B Foundation uses clinicaltrial.gov. Actually, anybody could have access to it. You get in that site and just put in the keyword hepatitis HBV, and we'll come up with all the clinical trials that's currently active. And some of them may be not particularly relevant, but I think that's one other way that you can stay abreast of at least any clinical phases of of drug development. And they often list many of the institutions and PIs, and whether they are in your areas and others. I think it's something that you just have to try to navigate. The site. It has a lot of information on there.
Thank you so much. That was actually the next question, was how do we get involved in clinical trials? So that definitely answers that. The other question that we have coming into the chat box, it says, it is a surprise to know that people under antiviral therapy with undetectable viral load are still infectious in animal study. How infectious are those under antiviral therapy compared to untreated people?
Well, it would obviously be much, much less infectious. This was an experiment not done to mimic their potential for transmission through the natural routes that human beings, that the virus spreads itself amongst human beings. It was a demonstration of whether or not it could be done. So the entire system was rigged to maximize sensitivity. A fairly large amount of human serum was transferred into the mice. The mice actually, in order to tolerate the human liver cells, have no immune system, so there's essentially no barrier at all to the virus getting started. So again, don't overthink that particular experiment. Its goal was to test whether or not persons with very, very low to nearly undetectable amounts of HBV or in their DNA in their bloodstream are producing actual functional virus. And it answered that question, but those data need to be limited to that question.
Just to expand on John's point, I think there have been experiments done in some of these infectious animal models, such as chimpanzees and woodchuck and duck. Virus, as few as one viral particle can establish infections. So as you can imagine, most of the therapy we have do not totally eliminate viral replication production. So even if you just have one virus, you still can establish infection. So I think which really suggests the importance of really totally eliminate any viral replications at all in order to achieve cure and also to prevent transmission.
Thank you. The next question we have, Chari and Sue, you both mentioned patients and their sharing of their personal experiences and living with hepatitis B and the direct impact that it's had on their lives, really daily lives, and impact stigma and discrimination and institutional challenges related with employment. Can you share a little bit about how more people might be able to share their personal experiences related to hepatitis B and how they might be able to get involved, get kind of engaged with some of the advocacy efforts that are going on to really push some of the awareness messaging?
Sure, I can get started. So we do have lots of ways through organizations to share stories. One thing the Hepatitis B Foundation has is the Just Be Storytelling campaign, and we also have the Be the Voice International Storytelling campaign. So if you go onto our website, you can see ways of reaching the Hepatitis B Foundation where you can share your story with us remotely. We actually have been doing these storytelling workshops in person. Of course, we can't do them now. So we do them remotely, but there are ways where people can create their own videos with us and can write down their stories, and then we work with people to help them learn how to more effectively share their story using social media. We can help link people with community organizations so that they can do more education and awareness in the community and help spread the word. I think there's a lot of different ways. World Hepatitis Alliance also has a Share Your Story, and they also have a network of community organizations around the world where you can link up with organizations near you. I think there's lots of ways to do it. We probably, again, if you go on our website or you share our social media, follow our social media, then you can see how to do that. But we also need to, I think, do a better job about making people aware of those opportunities.
Yeah, this is a great question. I think for so many people, as you hear a lot of these Just Be stories, it is a journey, right? It's a journey of getting to the point where you might be ready to share your story. And I find that patients, when they find each other, and I know with the Just Be campaign, when they get people together, like it really becomes its own support group as people kind of talk about their own experiences. And you actually, this has happened to me too, as I started getting more involved with the Alliance, the other board members I was on the board with who were all affected with either Hep B or Hep C, as we shared our own stories, it was very healing, but also very empowering for all of us. So I think there are online patient forums that Hep B Foundation has, and we'll try to start up another one when patients find each other and realize that they're not alone. That does help. The Just Be campaign is amazing. There's people from every demographic group, different situations. And so if you haven't had a chance, definitely go on the website and listen to some of these stories. They've had the storytellers come out to different events and share, go to the Hill. There are recordings that can be shared in different scenarios. We've definitely showcased some of them at some of our meetings. And the Alliance, we have something called the Wall of Stories. So if you get on our website, there's a way where you can post your own story. And we're trying to collaborate more, so we have a larger pool of stories that that anybody can access between different organizations and such. So, and our membership is all community-based organizations. So you can also see, depending on where you are in the world, that there are different community groups available, for patients to get plugged into. Thanks.
Thank you. And this is kind of a loaded question for anyone, I guess. How has COVID-19 impacted progress towards a cure?
Well, I think as I intimated that certainly with the COVID-19, the world's attention had turned toward this particular disease. And right now, I don't see any specific impact on the effort to develop HBV curative therapy. I think many of us still continue to work on it, including scientists, research labs. I think it's just the time of our world that the people are certainly much more interested and focused on COVID-19, which appropriately so. I'm certainly not ignoring that. It's clearly a critical, important problem, killing almost a million people in the world. So I think that's something we have to deal with it. But just knowing the virus, I think it's not going to last forever. I'm confident that we will have the vaccine. Certainly not by November 1st, but probably in the next year, and we will have good therapy. So I think hopefully once that's conquered, people still will be interested and focused on treating or developing a curative therapy. And because that's not going away, right? I mean, they're still going to have major public health problem in the world, and still millions and millions of people infected with the hepatitis B virus. So I think that's something that will continue to be an important public health problem in the next decade or so.
I want to second Dr. Liang's comments, is that this has been a more of a hiccup than a roadblock for us. So for example, my lab was shut down for 3 months. We have to wear our masks all the time when we're near anybody, but we're working hard. It's a hiccup, a minor slowdown. We see no decline in enthusiasm for supporting HBV through the NIH or anything like that. So this is a hiccup, not a roadblock.
Yeah, I think that's a great question. And we've heard that question a lot, people are asking, well, how does this impact people, like patients getting care, people living with hepatitis B? And definitely, like John is saying, it's a hiccup. We do think that there's been a pause on the progress to elimination in terms of service delivery for things like vaccines, for child infant vaccines and adult vaccines. In a lot of part of the world, people could not get it, and so there's been interruptions also in people getting their medications. And so, the patient community definitely, is going through some bumpy times. I think there are some positive things. So like telehealth, obviously, is something that is so widely accepted now and really does help with travel issues, and people can see their doctors through telehealth, and that could be a positive thing moving forward. And you've seen efforts of people really trying to get services to the communities rather than waiting for if people are unable to travel to hospitals, that the hospitals can actually distribute medicine through other ways. And so people have been creative. So I think there's a lot, we can learn from COVID. Thanks.
Yeah, I think we all realize that this is not the only problem we face in hepatitis B field, right? This is a system problem that all other diseases are facing the same issue of how to deliver the best medical care in this current condition. So I think this is something that we all have to get together in the medical system to figure out the best way to take care of patients, regardless what they have. So I think this is something that certainly goes way beyond what many of us taking care of patients with hepatitis B, that includes many other diseases. So I think this is something that will have to be dealt with at the system level, how to respond to these kind of a public health threat as with COVID-19.
Thank you all. The next question, what is your best prediction for a functional cure in the next 3 years?
Well, I don't play the lottery. Let's just start with that one. I do not believe that within the next 3 years, I mean, that is a very tight time frame for drug discovery. Typically, from concept to first approval of a drug, it is 15 to 20 years. We are obviously for many HBV therapies at the downhill slope on that, but at the same time, we've got to build our toolbox of drugs, which is what's going on. All of the trials that I highlighted, except the Replicor one, are monotherapies, meaning they're using one drug at a time. Once you have proven that a given drug is working, then you have to prove that it's safe and effective when combined with other drugs. That means more clinical trials. So within the 3-year time frame, I mean, I'm very hopeful that our functional cure rates will increase over that period, but I do not believe we'll be close to the finish line in that period.
Yeah, I would certainly echoed John's point. I think 3 years certainly not unrealistic in terms of having a curative therapy. I would just kind of want to maybe couch this whole questions on the whole conceptualization of the medical events. I think American people tend to have somewhat unrealistic expectation of what medicine can do. I mean, just to share a vignette with you, there was a survey done in 1950s about what people, all various different questions, and it's a population-based survey about what they expect. It's interesting that 70–80% of the people surveyed think we will have a cure for cancer by 2000, because that was done in 1950, whereas only 35% of people think we will go to the moon. Right? We all know what happened. We still haven't been able to cure all the cancer. We went to the moon many years ago. So I think people tend to have an unrealistic what medicine can do. I think it's really very important for us to relate that to others, that we are really, it takes time. I mean, that's the whole thing, that that's something we need to make really continue to articulate the fact that we have to do science. We've got to do good science. We've got to do safe science. We can't just rush things through because we need something right now. I think that's very critical, especially in this time period, right? We really need to defend the sovereignty of science and how this can be done in terms of developing treatments and vaccines, etc.
Thank you so much. I think a lot of it comes from a lack of understanding of the process as well from the general public. So maybe we can do a better job at educating our communities.
I think it's also tricky for Hep B because of the dramatic cures that we're seeing in Hep C. Yeah. So there are these expectations that, we're going to have the same kind of development and sudden rush of, cures that have over 95% cure rates. And so a lot of it is not understanding how complicated the Hep B virus is and how closely it interacts with the host immune system, right? So much of it is about the host response. And those are things that I think are important for people to, at least get a glimpse of. I don't think, most people will be able to understand a lot of the nitty gritty, but just at least some of the basics of how different the virus is, even though they're both called hepatitis, right? That they're completely different viruses.
Or even hepatitis C, even for hepatitis C, it took more than 20 years, right? I'm sorry to say, even for hepatitis C, it took more than 20 years until we have a curative therapy. So these things don't happen overnight. And I think that's very important to educate lay people that some of the things we will have to basically, it's a process. So.
Yeah, I agree, Catherine. I think we need to do a better job of communicating the process. People don't see the 20 years before the miracle, right? They just see the miracle. And I think, we actually get a lot more questions internationally about when the cure is coming. And I think there's the desperation out there is because people who are surface antigen positive can't get jobs, can't go to university, can't immigrate to other countries. I mean, I think the desperation is palpable, and I think there's some impatience there, and understandably, but I do think we can do a better job, to reassure people that there are scientists around the world working day and night, working really hard, that progress has been made. I think webinars like this, and we need to maybe do a better job of finding new and creative ways to get to communities that maybe lack this kind of technology to jump on a webinar, perhaps, just to let people know that we are working on it and we're working really hard, and that we need them. We need their voice to help prioritize hepatitis B too, so that there's funding for this work. But I think, I think an understanding of the process would help a lot.
Thank you all. This next question, for the HBV cure, is it solely the disruption of cccDNA, or would it be the simultaneous disruption of both cccDNA plus integrated HBV DNA?
Yeah, that's an interesting topic that there is actually a fair amount of debate on right now. The integrated versions of the HBV do not contribute to active viral replication. However, when they are integrated, in many to most cases, depending on which of the viral genes you're talking about, they can produce the viral proteins. And those viral proteins can be bioactive. The one that is the most important to consider in this context is the hepatitis B surface antigen, which can be expressed in significant levels from integrated HBV DNAs. And there is substantial reason, I don't think it's proof yet, but substantial reason to believe that the HBV surface antigen protein itself, just the protein, divorced from viral replication, is actually helping to suppress the immune system, leading to continuation of the infection. That being said, the infection itself is maintained by the cccDNA. So if you get rid of the cccDNA, permanently damage it, or permanently silence it, you will achieve a functional cure.
Thank you. The next question that we have, this is also for Dr. Tavis. It's a bit complicated, so I hope I can illustrate it correctly for you. How about the new immune modulator as part of combo therapy? For example, the first is DAA plus siRNA plus capsid inhibitor, or DAA plus siRNA plus immune modulator. Which one would you prefer?
Well, I'd need to see the data. I mean, to mimic Dr. Fauci, I don't have a horse in this game. I would look at the data and go where the data tell me, which means we need to have the quality clinical trials that Dr. Liang was referring to.
Thank you. So you mentioned the simplification of guidelines for providers. How can we work as a collective community to continue to promote the use of this to expand testing and management at the primary care level?
Yeah, we're trying to figure that out too. I think we do need more people in this space. I think, we've talked a lot about messaging this right within the physician and the provider community as cancer prevention, right? It's something that primary care providers do for breast cancer, for colon cancer, for prostate cancer. We do screening. So we think rather than people feeling already, even more overburdened by another disease that they need to get to know, at least just take that first step of screening. We believe if people start screening, and they get more patients that they find with Hep B, then they will be more interested in, oh, what is the next step? And, oh, this patient, I'm unable to find a hepatologist for this patient or a GI doctor who will treat him or see him, what can I do? And then they'll find that the resources are there. So I think we need to do some more bridging with other primary care societies like American Academy of Physicians with internal medicine, the family practice organizations. And then we've talked about, whether it's something that needs to be part of the quality metrics, to do, or it's at least recommended now by USPSTF to do Hep B screening in those at risk. But, we've talked about, should we really be pushing for universal screening like there is for Hep C and HIV? Because with risk-based screening, we are missing a lot of people. And when you look at all the people that are recommended in those groups for USPSTF and CDC, it's like this long list, which no medical student I think could actually, memorize. It's the majority of the population that falls into one of these risk groups. And so even with HIV, we recommend universal screening, even without risk factors. And maybe that's something we need to, think about to really get screening up there and diagnosis. And hopefully if we do all this right and we actually can eliminate, Hep B and and Hep C, then this won't need to keep going, right? I think we think of this as like a time limited thing. If we have we can vaccinate for Hep B, we can treat people, then in the future, it'll be less of a burden. Thanks.
Chari, do you have anything to add to that from the Hep B Foundation side?
No, I agree with everything Sue said, and at the foundation we have been, we're proponents of universal testing. I think, one of the challenges we see working with clinicians is how difficult it is, and if most people should be tested anyway, it's almost impossible to integrate risk-based testing at the health systems level. So universal one-time testing, I'd be all for it.
Well, perhaps this could be part of the universal screening, not only for hepatitis B, but also for hepatitis C, right? Because as you all know, there's been a lot of interest in have to expand screening for hepatitis C to a universal base because, again, with all the same reason you mentioned, that it's not capturing the patient with hepatitis C. So maybe, again, if you're going to screen people, why not screen several diseases that are going to be of relevance? So, right. So I think maybe that would be an area that hepatitis B field should work with hepatitis C, is a very different organization trying to promote the whole concept of a universal screen for viral hepatitis. Certainly, you could include HIV if people feel that's important. But it's sort of a simple, because you're going to screen people. Why not screen for a couple different entities, right? So
yeah. That's what I do now with my patients. Any new patient, I say, I do a panel, HIV, Hep B, Hep C, not risk-based. It's just, and people are fine with it. They understand you're trying to do a good job. I think COVID presents an opportunity too, because they're talking about how you can integrate that with other testing that should be done. So yeah, I think, we really need to think about person-based care instead of system-based care, and doing what's right for the patient and the full complexity, and really servicing that patient, in many ways, not just targeting disease by disease.
Great. Thank you all so much. And the last question I think we have, Dr. Liang, you had mentioned that the NIH had research dollars devoted towards research for hepatitis B cure. Is that enough? And what can patients and people on the line now do to help advocate for more research dollars so we can get to a cure?
Well, I don't think it's enough, but certainly I think has gone up substantially as the number I just shared with you. So I think it's going to the right directions. I would think that will require additional investments. And just sort of as a comparisons, like in HIV, they spend over $1 billion a year on research funding. And whereas HBV, although gotten better, it's only about $70 million. So there's a huge difference. And I think to have a curative therapy, I think there's going to need to be more investments, not only on basic science, but also in clinical medicines. And I think it really require a strong advocacy from everybody to highlight the importance of increased funding for hepatitis B. I think that's a very important, for instance, Hepatitis B Foundation has been very proactive in doing that. So I certainly want to give them a shout out for being able to at the forefront of advocating for curative therapy for hepatitis B.
And to put some of the cost in perspective here, the numbers are a little hard to come by because pharmaceutical companies tend to be a little tight reporting their expenses. But the estimate is to develop a drug from concept to first sale is typically between $1 to $2 billion per drug. And we are going to need a panel of drugs to routinely cure HBV in the wide spectrum of clinical contexts in which it's going to need to be done. And so that just puts it in context. There's a lot of money needed, so advocacy is for support for research is very important.
Yeah, I think as John mentioned that the pharmaceutical companies certainly have become very interested in curative therapy, hepatitis B, which is great. I mean, because they really have a lot of funding resource to do that. So I think this is another very important development because many of the pharmaceutical companies are putting this as a sort of a top priority, and which is going to certainly help to promote the field and come up with curative therapy. So that's another very important development in terms of funding for hepatitis B.
Thank you. Sue, we just had a question. I think this might be best answered by you. What are the chances of developing HCC for chronic hepatitis B patients?
So, I mean, the stats that you see out there are that 1 in 4 people living with Hep B will develop liver cancer or cirrhosis. You know, I don't know. I mean, I know compared to somebody without Hep B, your chances are much higher. I want to say like 50 to 100 times, and definitely other scientists, please feel free to chime in. But we do know that if you can suppress the viral load, you can prevent it. You can drastically reduce your chances. I think what makes Hep B different from Hep C, which is interesting, is that Hep C, the tumors arise from cirrhotic livers, whereas for Hep B, you don't have to get to cirrhosis to develop liver cancer. And oftentimes the very aggressive tumors actually happen in young people who have very healthy livers, which is, you know, obviously scary for us to hear that. But anyway, I'll pass that on to see if John or anybody else wants to add to that, or Jake.
I don't have much to add. I mean, you're absolutely correct. I think the important thing is to put people with high risk to cancer on suppressive therapy, and that can significantly reduce the risk of a liver cancer, but it doesn't totally eliminate it. We all have seen patients who are pretty good treatment, they still develop liver cancer. So the current treatment we have does not totally suppress or eliminate the risk for liver cancer.
Yeah, I think that brings up a really good topic too, Jake, in terms of, you know, right now the treatment guidelines are really very much based on patients having to prove that they need treatment. So you have to have abnormal liver enzymes. You know, your viral load has to be over a certain amount depending on if you're e antigen positive or negative. But we all know that the ALT, the liver enzymes, really go up and down, you know, and not everybody is going to be so closely monitored. I mean, I have a lot of patients who fall out of care, you know, maybe they get once a year. And so I'm often very nervous because I realize if they're on the borderline, there's probably some chance that I'm missing them when they're high up, you know, their viral load is, I mean, their ALT is high up. And so we do, you know, I think a lot of us are concerned that those of us who don't fall in the category for needing treatment, and I'm one of them where I'm relatively inactive. My viral loads like hovers between 1,000 and 2,000. I'm e antigen negative, so I don't follow the guidelines. But you know, we just worry. I feel like a lot of patients worry that they're sitting on a ticking time bomb, and this is why people get desperate and are buying all these, taking crazy herbal therapies, and they're looking for something. And so, I think we're hopeful that with the increased funding in NIH that there can just be more studies done, right? We see what happened with HIV and Hep C. They all went from treatments, where you needed to have a certain viral load for HIV or a certain viral load for Hep C before you needed treatment, but now it's treat all, right, for HIV and Hep C. I think when you talk to a lot of the scientists, they say there's not the evidence for Hep B, right? We don't want treatment to harm people. And even though it's relatively safe, are we potentially, I don't know, suppressing natural seroconversion? But I feel like the risk at this point, when you look globally, is that clearly we're under-treating people. Our rates of liver cancer are still really high. People cannot even get viral loads in Africa, right? How are they getting treatment and following guidelines if they can't get a viral load tested? We need to do much more than we are now, because following these guidelines, which are so strict, means that a lot of people are not getting the medicines that do work, because they can't get monitored. And the studies weren't done, right, in populations where people didn't have viral load. So, I think there's more and more studies looking at, how we can bridge those gaps, but it'll be good to have more research in that area.
Yeah, definitely there's a shifting prevailing attitude about treatment now. I think more and more people realize that maybe some of these treatment criteria we have set in the past need to be loosened, meaning that we may need to treat people who don't completely fit the criteria it used to be. And I think people start realizing that there are studies that are addressing these issues. So-called people sort of fall within the gray zone, may not quite fit the criteria, but they should be treated and they've been monitored in terms of their disease progression. So I have this feeling that, as you mentioned, once we have better treatments, if we do have a curative therapy, why not treat everybody, right? I mean, but at this point we don't. So I think that's why people are reluctant about treating everybody. There got to be a specific criteria that we should use to treat, and which I think will continue to evolve. And I think that has been actually for the last 10 years, if you look at the treatment guideline, it has sort of evolved in many ways based on science. So I do think that that's going to change, and with time, and people are going to realize that more and more of these people, as you mentioned, should be treated.
So I've got a question related to this. Tenofovir and entecavir came off patent not too terribly long ago, and so there's becoming generic versions of them available, which helps with the price barrier. How much is that helping penetrance of treatment into populations that may be resource limited?
That's a great question, and I can only speak anecdotally. The people who are reaching out to us worldwide and then in the US. Internationally, it's funny, I sat in on the COLDA conference, and I heard, I hear a lot that, now around the world you can get tenofovir for $40 a year and it's so cheap. We're not seeing that at all. It may be $40 a year for the government to purchase it, but by the time it gets to the patient, it's more like $40 a month in Ghana. And there's no way people are going to be able to afford that. So it's still what we're seeing from a couple thousand consults a year. Is that tenofovir, which is, I think, primarily the drug used outside the U.S. more than entecavir. We don't see much on entecavir. We're seeing tenofovir or even lamivudine. It is still really expensive in a lot of countries by the time it gets to the individual. So there's taxes and there's doctor fees and everything else. And so it still seems to be kind of out of reach for a lot of people in many countries. In the U.S., generic entecavir is more expensive than on-brand tenofovir. So that we're seeing with discriminatory drug pricing, drug tiering in the U.S., we're seeing that it doesn't matter if it's generic or brand, that we have people who now are asked to pay $1,200 a month for entecavir or tenofovir, depending on the insurance they purchase. So we're not seeing much of a positive movement towards accessibility and affordability.
Well, I think
John,
what we need.
Oh, sorry, go ahead.
I'm sorry. I think what we need is a Ryan's bill for hepatitis B and a PEPFAR in Africa for hepatitis B, right? I mean, those are the ones that really have changed the landscape for treatment of HIV, these national bills and effort and programs. I think that's what we need for hepatitis B, something like that.
Yeah, I think in terms of like, I think one of the ways we've really been trying to work, because I think it's hard to imagine that there's going to be anything like the Ryan White Act again for Hep B, and even for internationally, we're just trying to see if we get, with the push for universal healthcare and integration, we think there's opportunities where it can be integrated better even with HIV care. So for example, the Global Fund, which their target areas are HIV, malaria, and TB, can actually access tenofovir for countries at a very, very low price. Because tenofovir is used for HIV as well. But a lot of countries are not aware that they could actually buy tenofovir for their Hep B patients through a Global Fund. So I think there are opportunities to do better. I think nationally, domestically, and Chari and the Hep B Foundation were a really critical part of this when, with the drug pricing, just working on advocacy. There were letter writing campaigns because Vemlidy all of a sudden was taken off the CVS formulary. And so suddenly patients with no warning at all were unable to get their Vemlidy. And same thing with tenofovir when it became generic, patients had no idea what was happening. And all of a sudden their tenofovir, because of market exclusivity for the generic company that registered for it first, I couldn't believe it was like $100 a month for some patients when it became generic. So advocacy is super important. I was telling Chari she should mention Hep B United, so I'm going to pass it off to her just to talk about other ways that people can get involved with advocacy and even, potentially increasing pressure for Congress to fund more.
Yeah, I think that's part of this is, hepatitis B for a long time has been in the shadows. We all know that. It's a stigmatized disease. It impacts underserved communities around the world. We need people to start speaking up and joining. And I'm so glad someone asked that question about how they can participate. It's all about advocacy, sharing your voice and being loud, right? It's the squeaky wheel that gets the service. So we need people to be sharing their voice, sharing their stories. If you're having trouble getting your medication, email us, share your story, post it on social media. If you're being discriminated against, I know it's hard to share your story. It is. We know how hard it is. We'll help you. We'll walk you through it. But there are ways, you don't even have to share your story. There are letter writing campaigns you can join. You can anonymously even join some of these things. But if we don't get 292 million people, we should be louder than any other movement in the world, right? So we need at least a small percentage of people to just start joining our advocacy efforts around the world between World Hepatitis Alliance, ICE-HBV, Hep B Foundation, Hep B United. There are so many ways to do it. We just need people to join.
Great. Thank you, Chari, so much. And I just wanted to put in, since we're talking about, if you're a patient, if you're somebody living with hepatitis B, we have our email address here, as well as the website, which was referenced for accessing clinical trials and understanding different phases of drug development and where research is. So feel free to check those links out, which are provided here on this slide. In respectance of everyone's time, I want to conclude today's webinar by first thanking our panelists for their time and being with us today. This was a great discussion, and I think it was really helpful at providing a context to where we are in terms of progress towards a hepatitis B cure, and we thank you all for participating with us today. Thanks to our attendees for joining us and spending a couple of hours with us on the first day of October. We really appreciate you being here with us, and we hope that you will get involved with this movement to work towards a cure for hepatitis B and raise awareness and raise your voice with us in this movement together. So with that, we do want to let you know that this webinar was recorded, and we will be following up with everyone with the recording and an email that requests you to fill out a survey evaluating the webinar, the content, and speakers as well. So please fill that out as you exit the webinar. And with that, I'd like to thank everyone and wish you all a great rest of your day.
Thank you. Bye, everyone.
Thank you. It was a pleasure. Bye.
Bye, everybody. Thank you.
Bye.